Okay.
Got it. Thank you.
Okay, good afternoon. Welcome here. It's my pleasure to introduce Eric. I've been stressed for the last few days. How am I going to introduce him? But if you spend like a day with him around, like he puts this big smile on your face, so at least I care less now. I talked to a few people how they would best characterize Eric, and the unanimous expression is that Eric is my hero, and I must subscribe to that as well. So there are a few reasons for that, and so I will tell you a little bit about his background and a few things about him, and then we will proceed with the talk. So he went to the college at Notre Dame University, and for PhD, for graduate studies, he was at Yale. And there he started to work with Walter Gehring, and with him he went also to Basel when Walter Gehring was establishing the lab, and there he met Christian Nüsslein-Volhard. And they together started to work on Drosophila model and made a mutagenic screen which identified genes which essentially control the body plan and are involved in major important signaling pathways you can imagine in animals. Now, it sounds really simple and straightforward, but actually I would like that every master and PhD student here would be aware what it takes to make a mutagenic screen. That's one of the reasons why he's a hero. Because back then, you have to understand that basically all they had were Mendel's laws of heredity, Drosophila, their brains, and pencil. And that was pretty much about it. I'm exaggerating a little bit, but in fact they had to identify the first genes, and that was done with mutagenic screen, which is really a gargantuan work, because you have to screen tens of thousands of mutant flies, make the backcrosses, and from the screen came out a set of genes, which it turned out are the ones which are essential for establishing the body plan in Drosophila, and later it turned out that those are the genes which are, to a large extent, universal for establishing the body plan in animals. That's one of the reasons. The other thing is that Eric is a great mentor, and I think all the people who went for lunch or dinner with him at conference would subscribe to that his curiosity and really the passion for science is something what I would call contagious in best possible terms. I hope that the students today who went for lunch with him had also a lot of fun. He's also having great passion for promoting science, and that includes many different aspects. And one of them is, for instance, promoting in the American education system the evolutionary theory to be a proper part of the scientific curriculum, next to the other theories which are not scientific. So I think I said enough about Eric. I'm really glad that you could make it here. We have actually a small present for you. And I think actually the person who used really the laws of heredity is at the right place at the right time in Mendel's monastery, because he used the laws of heredity to discover these genes which regulate development. So I think it's the really most appropriate time and place to give you the certificate. And there is a medal. I hope it is in there, you know how. Yeah. So there is there's also medals.
Thank you.
Thank you. And the stage is all yours.
Okay. Thank you all for coming. It's amazing to look out and see so many interested faces, people interested in science. It's hard to come here to this particular site and not think a little bit about the history of science and the way science, the advances in science broadly speaking, affect our own development as scientists. The period when Mendel was doing his work was a period where the problems of inheritance were recognized as really central. He was not the only person who was doing breeding experiments, but he was actually he was the only one who saw to the heart of what the phenomena was. The period between 1880 and 1910 is, in my mind, the period where Mendel's work was rediscovered and reappreciated, is for me one of the most extraordinary periods in science, because the structure of cells, the idea of description of embryonic development, all of that was worked out in that period. And so you can look back in history, and you can always find extraordinary periods in science where ideas emerge and experiments and observations emerge. One other period, though, and I'm speaking personally, as a developmental biologist who's interested in embryonic development, the last 30 years have been an extraordinary time in developmental biology. If I think back to what was known when I was a graduate student, or when I was starting, about the mechanisms of development and what we know today, it's hard for me to even figure out or remember what I thought I knew back then, because so much has been colored by the molecular biology, the cell biology, the development. This is true for almost all the organisms that we work with. It's particularly true for flies, and that's what I'm going to talk about today. In most of these organisms, but in flies, we've learned quite a bit about how cells are directed to specific fates in the animal. We know in flies, for example, that the initial decisions reflect a pattern of distribution in the cytoplasm, of transcription factors, for example, like the transcription factor dorsal that is expressed in a gradient along the ventral to dorsal side. And high levels of this maternal protein elicit, this maternal transcription factor, elicit the transcription of two genes, twist and snail, that we've learned are the actual determinants for the mesodermal fate. Cells in an embryo make the choice to become mesoderm by expressing these two gene products. We're seeing here twist, but snail have the same. So we've been able to now understand cell fate choices and the programming of cells in terms of transcriptional responses of specific genes. These cells become mesoderm because they express this transcription factor. These cells become ectoderm because they don't. Now, what this slide also shows, and is for me one of the most remarkable aspects of the prospect, is almost immediately, and in flies within 15 to 20 minutes, after these genes are fully expressed, after these cell choices are made, the cells which make those choices almost immediately begin to change their shape, change their physical properties, and begin the process of in the case of mesoderm, of internalizing those cells to bring those cells to the right position in the embryo. So cell fate choices are somehow translated immediately into cell behaviors. Those cell behaviors, those shape changes, those folds, ultimately are a problem of mechanics, of physical forces in the response of cells to physical forces. So the exciting thing for me now, in having made it through the last 30 years, extraordinary years of developmental biology, is the prospect that what we've learned in developmental biology positions us finally to begin to understand the deeper aspects of cell morphology, the mechanical and biophysical aspects of how cells do physically what they do, how they move, how they change, how these cells, using the genetic information that we have that programs these cells, makes them different from these cells. We can ask, how does this play out in terms of physics? And so that's actually going to be the major idea, the sequence behind this talk. I'm going to focus exclusively on these mesodermal cells. And you can see here, this is a panel of actually fixed embryos, but I wanted to give you a sense of the morphological changes that the cells undergo. At the beginning of gastrulation, after these cells here have been programmed to be mesoderm, within about 5 minutes, the cells begin to change their shape, they begin to elongate, and then within about 10 minutes, a fold forms, and the cell, whoa, I have to be careful with this machine. There, a buckle and a fold into the interior of the embryo. It's about 15 minutes. It's not fast enough for you to see it as motion, but it's time lapse is a wonderful thing. So you can, and then, but interestingly, once these cells have moved into the interior of the embryo, they undergo a second major transition. These cells here, that up to this point were a simple epithelial sheet, a fold, they suddenly and simultaneously undergo a cell division. You can see that here, rounding up of these cells. And when they come out of that cell division, they've lost their epithelial character, and they begin to spread out along the surface of the ectoderm. They've essentially undergone what's called an epithelial to mesenchymal transition. They've lost the characteristic adhesiveness of epithelial cells and have become migratory. So what I'd like to do in today's talk, it really divided into two different parts. I'd like to spend the first part just talking about how transcription factors controlling cell fate control cell behaviors. Transcription factors are almost by definition in the nucleus. So, and yet the properties that we're looking at are outside. How is the rest, how are forces localized? How does the rest of the embryo respond? So I'd like to spend, and much of this work has been published, but I think it's worth going over it to give a background of our understanding. The second part, I'd like to address a slightly more complicated question. And that we've seen that the expression of twist and snail, the decision to be mesoderm, is elicited by these two transcription factors, twist and snail. But the process is complicated. There's a sequence of events. And yet these expression of these two transcription factors determine not only the fate of mesoderm, but they establish a sequence of events. So I'd like to ask how it is that these very different processes of epithelial folding, mesenchymal turn, are sequenced to ensure proper development. Because if something happens at the wrong time, then you're not, the sequence is actually an essential part. So I'll talk a little bit about sequencing. Why do the epithelials fold before EMT occurs, and whether steps and sequences can be completed. Now, these pictures just show a little bit, review the basic morphology that you can see here. These again are broken scanning EMs. If you draw the cells as cartoons, they start out as being essentially cuboidal. There's a constriction of the apical, or the outer side. The cells maintain constant volume, so a constriction of the apical side causes the cell to elongate. And then after about, depending on the temperature, 7 to 10 minutes, the cells suddenly undergo a second step where they shorten. And because they've constricted their apices and they shorten, their bases have to expand, and it's that conversion from essentially a square cell into a trapezoidal cell that produces this torque, essentially, that drives the internalization of these cells. This can depend on forces, and one of the initial cues or clues came from an examination of the major force-producing motor proteins in cells, cytoplasmic myosin called myosin 2, essentially the same variant of a protein that interacts with the actin cytoskeleton and produces contractile force. At the beginning of gastrulation, most of the myosin is localized to the base or inside of the cell, and you can see that here, and that's because of its role in a previous process called cellularization. These cells here are now expressing twist and snail, and what you can see is there's a change in the distribution of myosin. It's lost from being gradually lost from the basal side and accumulating on the apical side. And you could imagine that this putting contractile motors here would drive an apical constriction and could drive the actual cell shape change. Now, the myosin that we're looking at, the question is really, what is the relationship between this distribution of myosin and these transcription factors, twist and snail? The myosin is a protein which is supplied in the unfertilized egg by the mother. So the myosin, and virtually all of the cytoskeletal component, all of the basic cell biology is supplied in the egg already by the mother. But the local transcription or activation in specific regions must in some way interact with this maternally supplied myosin to change the distribution and allow non-uniform force generation. Ooh. Okay, yeah. So, again, and the interesting question is, these are transcription factors, and yet are driving maternal myosin. So one of the things in my own lab I spent a significant amount of is to try to identify or work out what this pathway is. And in end result, the pathway is kind of simple. These are transcription factors. In addition to controlling the general decision to be mesoderm, whatever that means, they also have specific targets, transient expressions of genes called folded gastrulation, T48, and MIST, which activate a Rho kinase-dependent process that ultimately results in the phosphorylation of myosin and its activation and accumulation on the surface. So you can see the expression of this folded gastrulation gene on the ventral side of the embryo. It's actually a secreted protein. It does not move very far, but binds to and activates the expression pattern. You can see myosin then, when folded gastrulation is secreted, you can see in response this accumulation of myosin here. The pathway, some of the components in the pathway are specific for flies. Others are conserved all the way up through mammals and humans. The pathway is rather similar, essentially analogous to the pathway that induces stress fibers in tissue culture cells. Now, what surprised us, though, was when Adam Martin and Matthias Kaschuber decided to look more closely at this myosin distribution that accumulated on the apical surface of this. I had thought that, well, if you have a cell and you want to constrict the apices, the simplest thing would be to have a ring of myosin around the outside, or some type of belt, and have that ring contract, and that I expected to see a distribution of myosin around the surface of the cell that would drive constriction. And instead, what they saw was a very different pattern. a pattern of myosin where you saw an arrangement that was basically corresponding to the grain of the cell distributions. But if you overlaid the positions of cells and the positions of in the mesoderm and the myosin, what you can see is that the myosin is actually a network, a complex network in each cell that's connected across all 800 mesodermal cells. So on the surface of these 800 mesodermal, they have a contractile network. The myosin, of course, in one cell doesn't extend over into the other cell, so there had to be coherent, like adhesive junctions that was holding this whole complex network. So what Twist and Snell does is establish this unified mechanical network on the surface of these cells. That was interesting in itself. What was, we know that existence of that network is expression of myosin is associated with a tension or a contraction. We can measure that using biosensors like the Ajuba GFP that goes to accumulates in junctions over tension. And what you can't see here very well is that the purple accumulation of myosin here on the surface is associated with an increased tension on the junctions. We can also use laser ablations and cut the cells and measure this tension. So the idea is that at this, what we've done is produce a tension, a network of tension on the surface of the cells. Now, that's all consistent with a kind of a simple mechanical understanding of what's happening here, that we're going to constrict the apices of these cells. What was remarkable in what Adam and Matthias saw was the second feature of this network that you can maybe pick up here in this movie. Now I go back forward, let's go again, and be careful here. What we're looking at here is myosin in green accumulating in individual cells here. And what you can, if you follow an individual cell, what you'll see is that even though this is a large interconnected network of 800 cells that locally there's a local activation process. So in an individual cell, you can see myosin accumulating, going away, accumulating, and going away. These pulses of myosin accumulation occur with about a 90-second frequency in individual cells. So the network is kind of locally activated and pulsing, and not particularly asynchronous with each other. This property, myosin pulsing contractile activity, is something that subsequently seen in lots of actin-myosin networks, and it's very interesting of itself what drives oscillations. That is interesting. Those are all interesting questions. I'm going to tell you I have no information on that at all, but the myosin pulsing was very useful as a useful tool. This is the typical thing that can happen sometimes in biology. Even if you don't understand something, if you observe it and think about it enough, you can learn things that you didn't necessarily anticipate. So we can follow here, and what we can see is that myosin accumulates in the cell, and as the cell myosin accumulates, we look at the shape of the cell, we can see that the cell becomes extremely distorted, and that's consistent with the idea that the connections between the networks are 90 degrees, perpendicular, so if you stretch the network, the cell gets pulled in this way. And then as myosin disappears in this cell and maybe begins to accumulate in this cell, the cell returns to its polygonal shape. But the apical surface has been reduced. So what this meant was that it wasn't only that myosin was pulsing, but that the cell shape change, at least the apical constriction, occurred in a stepwise fashion. And each step was coordinated with a pulse of myosin. So the morphological change, the apical constriction, generally cells would undergo 6 to 7 steps, and they would constrict down to nothing. And you could show by doing calculating correlation coefficients, that the timing is almost right on. An immediate response to myosin accumulation is this stepwise reduction in apical surface area. So this made sense to us, but it is an argument that the change in apical surface area is a response to myosin. We're going to use the pulsing and the correlation to say that it's a response. Now, the amazing thing, though, was that after we did this, we said, well, look at the cells. The cells are doing all kinds of complicated things. The apical surface isn't just restricting. The cells are elongating. Microtubules are becoming long. The nucleus that used to be here is moving here. All these things are happening in the cell. And we initially expected that there could be all other kinds of cell biological machines that would be driving those things. But each of these processes, the increase in the surface area of the cell, every one occurs in a step-like fashion with a 90-second periodicity with a correlation that is exactly similar to what we see with apical constriction. So that has led to a view that myosin does everything. That somehow, and the challenge we have to face, is that a pulsing myosin machine drives a morphological transition that, and maybe we can begin to think about this, accounts for much of the morphological transformations. That problems in biology might be simpler than what we would be led to believe just simply by. So I'm going to pursue that idea for a little bit. We'll come back over the course of the, to how much can we attribute to myosin. It's going to be, what you'll probably end up with is, this guy is a crazy myosin fanatic because he believes myosin does everything. And I may not actually believe that, but I'm going to argue for that. Because I think it's very useful often in science to argue, to push an idea as far as you possibly can. Okay. But I'll give you a couple of examples, and I'll let you, so remember there's this morphological sequence. Cells elongate, and then they shorten. We've been talking a lot about all the different nuclear display elongation, but what about later changes? And so we started thinking about this mechanically, and Oleg Polyakov, who was a graduate student in physics who had joined, was building little computational models for ventral force, essentially vertex models where you treat cells in 2 dimensions as rectangular, or that were connected by vertices and would have elastic properties connecting the points here. What he asked was, since we knew that the cells maintain constant volume, if you squeeze the top of the cell, I'm going to do this like not a physicist, I'm going to do it like me, and Oleg, I can tell you, did this computationally with and could make, but I'll give you my intuitive understanding, because very often that's the way it is. When you finally understand what the physicists are telling you, it is kind of intuitively true. And so if you squeeze, the consequence of squeezing apically will depend on 2 things. It'll depend actually on how stretchy the lateral surfaces are versus the basal surface. If the lateral surfaces are weaker, then a cell will elongate, obviously. If the lateral surfaces are more stiff, the constriction and the change in the shift in cytoplasm has to be taken up by an expansion of the base. So you have one of those 2 alternatives, and you can look at these 2 morphological transitions. You can get a fold here. This is what physicists would call a buckle. It's and you have this thing here, and you can look at both of them, and none of them really look like a Drosophila embryo to me. That's what I told Oleg. And then Oleg, said, no, wait a minute. We have this sequence, and maybe the first part is this is characterized by a softness of the lateral membranes, and then the second part is more similar to what would happen. And so then what Oleg suggested was that, well, maybe we can understand the whole aspect here, this additional next step, as a change in relative stiffness, a transition. So initially, you do chemical work by myosin ATP hydrolysis. You constrict apically. Part of that work is to extend elastic springs, which are laterally displaced, because they are the most stretchable. But if those springs are truly elastic in any sense, they store some fraction of the chemical, of the work of that myosin acting as potential energy in an elastic, in a spring. And if the basal surface were to become increasingly softer at some point in the process, the calculation for the cell would be expand the base. The lower energy configuration is to expand the base and contract the lateral surfaces back. And then quite remarkably, what we did was, like, if you measure the loss of myosin, and this loss of myosin, basally we showed genetically was different than the Rho-induced activation apically, but there's a different process that provided a clock. Essentially, you had myosin lost at later and later stages until, in fact, apparently there was a stage that was consistent with shifting the appropriate distribution and driving the invagination. What's interesting is that this elongation, the cells become long, and then they shorten during the actual invagination, and they shorten back to the same length that they were before. So this is kind of like a spring. So I love this model. I think it's great. This elastic, the response to elastic stretching. My friends who are cell biologists tell me that this is the most ridiculous idea they've ever heard. Because they think, what are these elastic structures in cells? Where are these magical springs? And I say, well, the cortex, the cytoskeleton, I don't know, something like that. And they would say, do you know anything about the cytoskeleton? If you do a FRAP experiment, and you look at restructuring of the cytoskeleton, the timescales are on the second, 2 to 3 seconds. So if you have a spring, and you are remaking this, you pull the spring out, and you remake all the little chemical bonds in the spring every 2 to 3 seconds, and you release the spring. Well, we all were children at one point, and we had toys that depended on springs, and we know that in the non-ideal world of physics, springs restructure over time and have a lifetime and only retain their ideal length over a certain period of time. And so the real question then becomes, are there meaningful biological elastic structures in biology that would have lifetimes of 4 to 8 minutes, that would be, and that was a good question, but it's an interesting one, because what you'd like to know is, can you do work? Another way of rephrasing this that I think is theoretically much more interesting is, can you do work and save some of the energy of that work for some later morphological process? That's really what a spring is. Generally in biology, you will be told, and I would have to agree, that mostly we describe everything as viscous. What that means from a practical standpoint is that whenever anything happens, it happens because you put more energy in. And you do this, and you move something, you constrict something, you produce a cytoplasmic flow. You don't save any of that energy. It all goes off as, essentially, in a viscous system as heat or friction. So we set out to try to test that model by saying, can we learn anything about elasticity in embryos? The strategy that we used, this is Konstantin Dubrovinsky, was to inject beads, fluorescently labeled beads, into embryos. And then we were doing this for a variety of reasons. We like to watch the beads when they were, watch the beads, how they jiggled, and try to calculate viscosities. And, the various physical kind of things that you would do if you had that. But Oleg and Konstantin also built little electromagnets that were small enough that we could put under the microscope. And as you can see here, we could bring the magnet in and distort and pull on this, and then pull the magnet away or turn it off, and then ask if biological structures were fully viscous, you would pull them, you would displace them, and they would stay where they were. But as you can see, obviously, in this image, you can, it springs back. If you watch, so we can do this more quantitatively. This is a chymograph where we're pulling and then we spring back. The shapes of these curves that you can calculate here are interesting because they already tell us something about the system, and they provide us the quantitative data that Konstantin is going to use to calculate. So what you can see is that if you pull something, a constant force through a viscous fluid, it just moves at the same rate. And you can see, whereas if you pull something that's on a spring, you can see that it begins to slow down. That's the characteristic of the spring can be measured in two ways. That here, as you pull, there's the shape of this curve allows you to extract a lifetime for the spring. But the other, an easier way, is to ask if you release and you come back, and you don't come back to the same point, that gives you a measure or potential measure of the lifetime of the spring. And the calculations, oh my gosh, oh good, okay, good, go back. And I've left off the math here, but essentially when you do that, what we find is that this system detects elastic elements that have lifetimes about on the order of 4 to 8 minutes. And the difference here is largely how you model the system, whether you model it in one direction as a linear spring, or whether you model it as a connected set of springs where some of the failure to come back is due to what's called the floppiness of the spring, the ability to reorganize elements without actually. So what this tells us is that there are these elements. What we still don't know is where they are, what they are. We don't have good models for how to relate the properties that we're measuring here to any particular cytoskeletal elements. We do know that if you disrupt actin, we lose all these properties. If you disrupt microtubules, you don't. We don't know, though, and these experiments are very specialized because we're doing them, actually, we're looking at the cortical actin, and in Oleg's model, we actually have not been able to figure out ways of measuring locally spring elastic. So all that I can tell you is that the model, attractive as it is, it may not be totally ridiculous, but we haven't figured out a way of really measuring or proving it to be true because we don't have ways of locally measuring elasticity. So the last, a myosin regulation story. So okay, that's ventral furrow, but let's talk about the whole embryo. There's a lot more to the embryo than just ventral furrow. So look at this guy. He's doing all kinds of very interesting things. Is all of that due? Can we explain all of that through myosin? And this is where it gets a little scary because it's a much harder problem. We've established a collaboration with the Boris Schreimann's group and the Kavli Institute for Theoretical Physics at Santa Barbara, and this is Boris, and some the major players, Sebastian Streichen, Nicholas Knoll, and Matt Lefevre, who's a graduate student in my, and this is, me, you have to remember, I'm sitting here with a camera, and I took this picture, and what you can't see, Boris is explaining something, you probably can't see the somewhat confused, furrowed eyebrows as we're going through some deeper, Matt looks much more thoughtful than I am. I'm fortunately out of the picture because I'm taking this. Okay, so our idea, and this is I'm just going to give you a plan of what we're doing before moving to the second half, is to quantify the distributions of myosin over the whole surface, 360 degrees over the entire embryo. Maybe if we have to identify other force-generating systems, we're going to start with myosin. We want to then, in living embryos, we want to build quantitative maps, what we'll call flow maps, which are basically distributions of morphological change. How cells move over the whole surface, over the entire blastoderm surface, and then take these two quantitative real measurements and somehow couple them to a theoretical model where you have the magnitudes. Can we predict the flow patterns based on the distribution of myosin? And then we can do that in wild-type embryos. Can we actually use mutants in flies to test whether these, the universal applicability of these models? So I'll just give you a little bit of a sense of where we are. Myosin quantification, we can do living embryos. This is a panel where we're looking in cross-section at an embryo, and we're looking also simultaneously at the apical and basal surface. This is the accumulation of myosin on the basal side of the embryo, and this is this basal accumulation of myosin. Here you can see it getting lighter. You can also see apically myosin, as cellularization is completed and gastrulation begins, you can see myosin accumulating on the apical side. I think it's a spectacular movie. Bing He made this movie. What we can do is quantitate these levels. I'd point out to you that in these early stages, the myosin distributions are structurally different basally. They're like interconnected rings, and apically is this network. But in both cases, the myosin distributions are essentially unpolarized. That is, if you drive a contraction of a local area, here the contraction will be isotropic in all directions. A little bit later in gastrulation, things become slightly more complicated because as myosin accumulates on the surface of cells, the myosin has a bias along anterior-posterior interfaces, and this is thought to have to do with process of germ band extension where individual interfaces are lost in the process of intercalation. So being aware of all that, what Sebastian had to do is to develop strategies for not only measuring myosin everywhere, apically and basally, but also characterizing the anisotropy of the myosin distributions. And there are computational strategies to do that. The other aspect of the project has been to use light sheet 360-degree reconstructions of embryos during gastrulation to follow the behaviors of all cells, and then to develop a computational strategy for plotting out what these movements are by essentially opening up an embryo and spreading it out into 2D. So we can define flow patterns on the surface of the embryo over a 20-30 minute pattern. And this is the kinds of flow. Initially, the cells seem to rush towards the dorsal side, and then as myosin accumulate, they move to the ventral side, and then there's this complex vortex movement. What we can do is now compare these flow patterns with the distributions of myosin. The early sequences, you can see that when myosin is rushing towards the dorsal side, actually on the basal side of the cells, there's a high concentration of myosin dorsally, and then this shift in the pattern is associated with high accumulations of myosin on the ventral, which is the outside side here, and then a more complicated pattern here. Computationally, what we've done is used these values plus the anisotropy for at every point along the surface of the embryo to calculate a measure of myosin that we're going to call a myosin tensor. It's essentially a value that has an intensity and a directionality to it that defines myosin at each of these points. And then the theoretical question has been to ask, if the myosin tensors are uniform over the whole surface of the embryo, there's going to be no movement. But if you have a discrepancy, any discrepancy in the intensity or the directionality, the character of this myosin tensor, that difference will predict a stress and will therefore predict a flow. And you can use those, therefore, the differences to computationally predict flows. And you can't, well, let's step back a second. We don't measure forces. What we measure is some magical myosin intensity that we think is somehow linearly related to the forces that are generated. But even if we could measure forces, that wouldn't predict flows. Because the other property that you have to all the other properties in the embryo. Something you can think of as friction or viscosity or just knowing that wasn't, but the question we've been able to ask so far is that can one explain the morphological transformations by these myosin tensors if one assumes a single value for this resistance, this viscosity? Or do we have to invoke changes in softness or other properties to change how, to explain how the cells are moving? And the answer is that so far, with some exceptions, if we compare the predicted flows with the measured flows, we are at about a 90% accuracy, which is very close to the accuracy that we're able to measure things with. So we think that essentially this is true. The only thing that matters is the distribution of myosin. You do not have to invoke other properties like softness or adhesion or changes during these early events. There are places where we have trouble until we have to go back to the models and introduce the simple models. And initially the models were developed with having no place for the cells to go. So if you'd have local contraction and local flows, but everything had to stay on the surface. And that gave us a terrible problem during ventral furrow formation where cells leave the surface. And so you have to artificially manipulate your model to get rid, to allow cells to flow out. And then when you do that, you can achieve again this 100%, this 90% accuracy. So this is kind of very promising and exciting for us. We're still struggling with the grain of our descriptions, but it means that at least in these early stages, following the regulation of myosin may be enough to account for and actually model all morphological change. And we're at a point now where we're beginning to test that model by changing mutants, by looking at mutants. What I want to do is go quickly through this next question, which deals with a slightly later stage. And we can be at the end then ask, does that really relate to this myosin-centric model? You remember the cells fold and move into the interior, and then once they're into the interior, they undergo they divide, undergo an epithelial mesenchymal transition, and then spread out over the surface of the embryo. Now, most of our stuff, work on flies, has been, we've always avoided this problem because it happens inside the embryo, and it's hard to image. And it's hard to image deep, it's hard to get accuracy. And so that's why we're very selective. We'd only do the easy things. about a couple of years ago, I had started deciding that it would be potentially really useful to try to understand these morphological transitions. And so I wanted something with the highest possible resolution. So I decided to go back to fixed material and scanning EMs. And what I wanted to do was to fix the embryos and then, that lets you look at the surface, but fix them and then dissect them, remove the yolk, and mount the embryos in a way where I could look down on the cells on the surface. And I wanted to do this in a way that I could follow mesoderm cells and mesoderm behavior. This is the only experiment that I actually did in this talk. So that's why I'm going to spend 5 or 10 minutes on it. All the other things that people are talking about, I'm going to zip by in one slide, but I'm going to waste 4 slides on this experiment because it's mine. Okay? Okay, so it works. You can, what we found is that lac-Z staining is compatible with the, it's the only, the actual excal reaction. The only thing that's really compatible with glutaraldehyde cacodyl fixation. So here is an embryo which has formed the ventral furrow, has moved into the interior, and I've pulled away everything else. This is an embryo where this ventral furrow cells are now dividing, undergoing mitosis. And this is one where it's spreading out on the surface. This is the EM. I have to say, I first have to photograph the embryos and then coat them with metal for EM. So we have these 2 images, and you can see the cells beginning their spreading like behavior, and you can kind of superimpose the 2 images. So I know that these cells are mesoderm in their spreading behavior. And it's interesting now to compare the mesodermal activities. One of the things that you'd like to know, we have this sequence of events. Normally the cells divide and then they do this spreading. So can we eliminate the division, which is controlled by, in flies, by the CDC25 phosphatase? And what happens if we eliminate this division? Is the division an essential property of the EMT? And I can tell you that there are mutants in this gene called CDC25 fail to undergo all post-blastoderm mitosis. This is a tool that we have available to us. The embryos don't make 6,000 cells, and then they amazingly try to go through the whole rest of embryonic development with those 6,000 cells because they can never divide again because they can't express this CDC25 gene. So when you look at them, amazingly, these big cells that haven't divided spread out over the surface and show essentially the same types of behavior. So we don't really know. So this is an interesting result of itself, but in a way, the way I'm thinking about it is that it's potentially more of a tool. Because the moment you say that mitosis isn't necessary, then if you're interested in the EMT, epithelial mesenchymal transition, you want to be able to see it and not the mitosis. And so I think understanding what an EMT looks like in this case, in the absence of mitosis, is very powerful because all these cells simultaneously within 3 minutes do this thing, so I want to know what it looks like. And that's the goal of this particular project. That's what I'm doing. I'm going to enter in. The other question, though, is that this EMT that one sees in these cells, this epithelial mesenchymal, in a way is not surprising. I told you that mesodermal fates are driven by twist and snail that establish this contractile myosin. But snail, for many of you, you should know that the snail gene, although it was initially identified in Drosophila, its major role is thought to govern EMTs in vertebrate embryos. So the snail homologs are associated with EMTs, and in fact, not only twistons are these 2 genes that set the cell off as a major regulator of EMT, and also they play similarly roles in metastases in cancer. So that this expression of snail is what is driving the EMT. But we have here now what for Mo Wang was an interesting problem, that the same snail expression that drives epithelial folding also drives the EMT. But the embryo, from a practical standpoint, has to ensure that if you wants the EMT to occur only once the cells are inside, it has to ensure that somehow epithelial fold happens first. And our initial sense was that, well, maybe snail comes up and is a transcription factor, and low levels of snail may induce epithelial folding, and then you could continue to increase the levels, and ultimately you would get reach the levels required for an EMT. So it would be you could sequence events by coupling them to the expression levels. And Mo checked that possibility, and turned out it was wrong, but wrong in an interesting way, in that actually, if you looked at the junctional disassembly and you followed snail expression, what you saw was that junctional disassembly in mesodermal cells begins even before gastrulation. The moment you have snail there, the junctions start to be taken apart, and the epithelium, you can't quite see it, you can see it measured here. There's a period where the cells appear to be entering, specifically the mesodermal cell, entering an EMT. And then the moment gastrulation begins, the junctions are restored, and the cell backs out of an EMT and continues the epithelial folding. And so you can follow that a little bit here. This rescue of junctions is temporarily correlated with the presence of myosin. Remember, this apical myosin that's driving the junctions are being induced to go away by expression of snail, but this myosin, as it restructures the junctions, drives them a bit apically, and you can see that maybe here. This is myosin, and this is the junction moving up. You can change the rates of myosin and change the movement and intensity of the junctions, and you can show that the restructuring of these junctions, this rescue of this junction, temporarily increases of, say, intensity of the junction, correlate with myosin pulses. So that led to the idea that EMTs, epithelial mesenchymal transitions, that are induced by snail, which is the standard thing, the snail, whatever it's doing to drive this epithelium, is sensitive to tension, to myosin tension, that cell cannot undergo an epithelial mesenchymal transition if it's under tension, is a way of saying that idea. So to test that, and that seems to be true of mesoderm, but mesoderm is complicated because, of course, we're programming these cells to... So what Mo wanted to do was to develop an ectopic epithelial mesenchymal transition, and then ask if we can induce this epithelial mesenchymal transition, is just supplying tension back enough to suppress it? And so her idea here was to use the fact that mesodermal fate depends on the expression of twist and snail, two genes. And if you eliminate one, or if you eliminate the other, the cells do not become mesoderm. So you have to have both to be mesoderm. Said differently, the ectodermal cells are ectoderm, and if we express either twist or snail in those ectodermal cells, they remain ectoderm. So what Mo set out to do was to take a normal blastoderm, where you can see these apically localized taut junctions in the mesoderm, and then these broader junctions in the ectoderm, and this is in the presence of snail, which is inducing both EMTs but also apical cancer. And here, what she's going to do is ectopically express snail over the entire embryo. And what you can see is now in the ectoderm, that expression of snail is able to disassemble the junctions here. So she's artificially inducing by ectopically expressing snail, inducing in the ectoderm an epithelial mesenchymal transition. But you notice here, the junctions are still here, because this is mesoderm and it's under tension. So even the ectopic expression of snail that disassembles junctions here is not able to do it in these cells because we're expressing twist and snail and have an active. And so then what she did was take these embryos and added an additional component, activated the gastrulation, folded gastrulation pathway that drives apical myosin accumulation. And so now we have UAS, you have snail-driven everywhere, but also myosin contractility everywhere. And what you can see is this tension that's induced by a UAS rescues these junctions in the ectoderm. So for us, this is an argument that two things, it points to two things. One is that snail may only be able to induce disassembly of junctions that are not under tension. And I think that's structurally interesting. We'll talk a little bit about that in a second. But that also, that potentially this sensitivity to tension is what controls EMT timing. The embryo activates both these pathways, but one pathway physically or mechanically trumps the other. And until the apical surface constricts down to 0, and until myosin is effectively lost, these cells cannot take their junctions apart. Now, what I'd like to do in the last 5 minutes is just discuss where we would like to take this observation. So how does snail expression, this is a fundamental question for all EMTs, how does snail expression drive an EMT? Why in neural crest cells, when you express the vertebrate snail homologue, do these cells delaminate and undergo an EMT? And in the simple case, and the conventional mechanism, the one that's been well characterized in vertebrates, is that snail is a transcription factor, is a transcriptional repressor, and it represses the transcription of E. cadherin. And we know that's the case substantially. What we know is that for snail to suppress or disassemble junctions using this mechanism is going to depend on the lifetime of cadherin and the turnover rates of cadherin. So unless snail interacts directly with the junction or there's some more direct process, this is going to depend on this transcriptional mechanism isn't very attractive. What makes it even less attractive, and actually impossible, is that the cadherin proteins that are being disassembled are maternally supplied cadherins. Like all cell biology, like all cytoskeletal components, it's all maternal, except for these transcriptional regulators. And so snail, the cadherin that's being lost, is not going to be sensitive to cadherin transcription. So we have to have some post-transcriptional mechanism, some way of directly affecting the cadherin that's in these junctions. And it turns out not just to be cadherin, many of the junctional proteins, and as a complex, are downregulated. And the one that we're particularly interested in, because we believe that it is actually upstream of cadherin, is a protein called PAR3. PAR3 in wild-type embryos is lost from the mesoderm. In the absence of snail, it's maintained. It ectopically, snail downregulates PAR3. And most interestingly, downregulation of PAR3 downregulates cadherin. So we think that maybe the real target of snail, in an immediate sense, is PAR3 and the structuring of the junction. So what we want to do is find out how snail affects PAR3. I'm going to give you our two strategies, and then we'll One simple strategy, model one, is that snail, which is a transcriptional regulator, downregulates the expression of a mysterious gene called gene X that's not supplied maternally, and gene X then is an active protein that interacts with the cytoskeleton and downregulates cadherin. And we have never identified a gene for that. We have other types of genetic screens in the lab where we eliminate whole chromosomes and can follow. I don't want to go, but we have actually begun to identify some candidates where if you delete the whole X chromosome but to supply back all early genes, you would show that there's a failure to downregulate PAR3. And so this is an interesting model, one that may most likely be correct, but we're also beginning to entertain another model, which is, I'm presenting to you in a little bit embarrassed state because I think it's very exciting, but it's probably wrong and certainly controversial. And you remember, in the opening part of this talk, I showed you a model called an effort to measure tension in junctions using a protein called a juba, which binds to junctions under tension, where cadherin, so it's a component of, functions as a component of the junction. A juba, even though it does this wonderful thing in flies of binding to junctions under tension, appears to have another role in vertebrates and was initially identified as a co-transcription factor of snail. And so in this model, from one of the original identification papers, a juba binds to snail and participates in the downregulation transcriptionally of cadherin. So we don't know that this is true. We don't like transcription regulation models. We don't think that downregulation can account for what we have. But the idea that snail as a transcription factor binds to a protein that's present normally in junctions suggests the possibility that there could be a more direct interaction between the expression of snail and the disassembly of a junction. And we haven't really been able to test that, but we have crappy antibodies to a juba. Looking at a juba localization, you see it's generally in junctions, but you can see right immediately before the EMT that a juba begins to accumulate in nuclei consistent with the physical interaction with snail. So we're beginning to entertain the idea that maybe what snail is doing is titrating away a juba or other, that there could be other physical properties. And so we're kind of fighting our way through the different models. We have some candidates. The candidates so far give us uniform expression. They seem to be essential for the process, but they're not behaving as though they're snail targets. So we're kind of stuck in the figuring out what the final what the actual appropriate models are going to be here. So I think I'm going to stop there. Actually go back. I wanted to end on things that we don't know and things that we're speculating with. I think we're in an exciting time here because in a certain sense what we have is a process that an entry point into a truly fundamental process in biology, this EMT, that comes a little bit from the genetics and comes from in an unexpected kind of way. And that the opportunity to maybe understand that these observations, we hope will give us a way of understanding what's really involved in the EMT, that we can then begin to extrapolate to be useful for people who are working on EMTs in other invertebrate systems or even in disease conditions. It's also interesting to us that, getting back to our myosin-centric work, that in thinking about what a protein does, we can think of the immediate effects of making a contraction here and say, in the types of more models that Boris Schreiman is working on, that you constrict here and your local differences in tension predict flows. But we think that tension may be able to do lots of other things in biology, and we have to begin gradually to begin thinking about what those possibilities are. Okay, the work, our work on gastrulation is a wonderful collaboration between this cell biologist, experimental biologist in my own lab, and a biophysicist in the Sigler Institute. In particular, is established by Adam Martin and Matthias Kaczube. I talked about Oleg Polyakov's work and Michael Gelbart's work, who did much of our cell tracking stuff. Currently, it was being carried on forward by Bing He and Konstantin Dubrovinsky. Mo Wang took a separate path and it really deserves, except for my little skimpy scanning EMs, deserves all the credit for the whole, the last part on EMs, and on EMTs, and I'll stop there. Thank you for your attention.
So I think, I don't know what are the rules of the house here, but I guess it's time for questions, right? So are there any questions in the audience?
It's a very hot room. And you guys, I'm up here with more air between me than the rest of you, so you, yeah.
There is one question here.
Question? Yeah.
Well, thanks for nice. It was quite a complicated talk. So if I understood it well, so the message is, so the snail is necessary to kind of release the junctions between the cells, induce the myosin, which then kind of recovers the cells together, leading to the specific changes in the cell shape.
Yes.
And all these events are leading into the EMT, right?
Right.
Did you try to express myosin ectopically? In other words, is that myosin expression sufficient, or at least partially sufficient, to induce the EMT transition?
Okay. All that we've been able to do is we'd like to, the question is, I think, is myosin expression sufficient to block EM?
To induce it ectopically, if you ectopically express myosin without snail.
So what we see if we topically express or activate myosin, say, using the folded gastrulation pathway, what happens is you induce apical constriction, you do not induce snail, and you do not induce an EMT. So the sense is that the folded gastrulation pathway will activate this myosin contractile pathway, but that can occur, conceptually, that it can occur independent of the EMT. It could occur in cells that were not undergoing an EMT. Now, the deeper thought, maybe, behind the question is, do we really have any instances in flies where we ectopically draw, or that folded gastrulation drives myosin accumulation that are not followed by an EMT? And I think there are a couple of cases, but it's really worth looking at them in greater detail. Almost all invaginations in flies involve this local myosin induction, and almost all of them involve expression of folded gastrulation. Not all of them produce obvious EMTs, but we haven't looked in detail. And it'd be useful to look at the junctional, the effects of the junctions. Yeah.
Okay. Other questions? You can shout.
Thank you. I'd like to ask, during the epithelial mesenchymal transition,
Yeah.
this stage, so the cells that are going to become mesoderm, do they have properties or characteristics of a mesoderm before they start to bulge, before they start to transition? Because based on the expression of the snail, it would seem like they are somewhere in between, or maybe they are already.
I think it's an interesting question. What we see is there is a loss of, the snail is associated with a loss of junctions. That's one mesodermal property. We have done transcription profiling and ask how many genes are regulated up and down by either twist and snail. And unfortunately, it's in the range of hundreds. What we haven't done is those experiments with a careful enough timing to say that these genes which are being controlled, these mesoderm-specific genes, are expressed. I'm fairly sure, I would bet you that they are expressed before the EMT and during this period. So snail is, so they're becoming mesoderm, but they're kind of, if you will, they're kind of stuck in an intermediate state.
Yeah, they're like blocked.
They're blocked in, and that it is in a certain sense an interesting state for cells, not to be totally mesenchymal, but to be in the process of disassembling junctions in a kind of an intermediate EMT. I know that Mo, who is just taking on an assistant professorship job in Nevada, is one of the issues that she is really interested in is the nature of this state of cells that are, on the one hand, entered into an EMT but not allowed to complete, not allowed to complete this. And can you keep the cells in that state? And maybe that state is more analogous to what happens in cancer and in metastasis. And we don't know, and they're interesting options. Yeah.
Because from your earlier pictures, it seemed like the cells start to becoming mesoderm and then they are blocked.
Yes.
And then it later finishes. Like,
Yeah, exactly.
Did
And so what is that block state? It's not going back to zero.
Yeah, but I mean more like, from evolutionary point of view, it seems like it evolved to, like, the mesoderm evolved sooner than the block.
Yeah, right, and that's true, I think. Huh.
Thank you.
Okay, thank you. So,
In the first part,
yes, I can.
in the first part of your talk, you proposed that the myosin distribution is sufficient to explain how the cells will move and how the shape will change. But what controls the myosin distribution?
Yeah. So we know what controls the myosin distribution in the mesoderm, twist and snail. We know what drives the anisotropy of the myosin in the ectoderm, and that's actually the juxtaposition of cells that are different to each other. All those are transcription factors. And so there's a thing that we don't fully understand. It's always still we know that in the blastoderm stage there are an extraordinary specificity of different cell types that are defined by different transcription factors. Somehow, those transcription factors then impact the distribution of myosin in a variety of different ways. It's probably different for each of those cell types. So even though we've, in a certain sense, solved the problem for mesoderm, because we know that twist and snail activate folded gaster, we don't really understand it for the whole rest of the embryo. We assume that it has something to do with the transcription distributions of transcription factors and cell fates, but we don't know the mechanism. Maybe for only in one other case for the anisotropic myosin distribution, this is related to the parallel-driven expression of receptors in the Toll family which activate myosin. So there's a pathway there, but many more pathways, and we don't understand.
Okay. Other questions? I would have one, maybe very naive, but in a myosin-centric world, could actually myosin become, say, a druggable target for cancer therapy of some of the cancers? Could you envision something like that?
Yeah. Again,
I mean, there are some cancers which are metastasizing, right?
Yeah, this is a wonderful week to be talking about cancer therapies. And for me, the feature, it's more the feature of an EMT which is revealed by its sensitivity to tension, is potentially provides an opening to think about how one might affect EMT. Overexpression of myosin or underexpression of myosin does not seem like a good strategy to target tumors. But I think what we've learned from this year's Nobel Prize in Medicine, Allison and Junko were not initially interested, I think, in curing cancer. They were interested in the complex interactions of T-cell activation and inhibition, and maybe they thought what they were doing would be useful for studying immunities or the immune system, or maybe even autoimmune diseases. But interestingly, its major usefulness and impact has come in terms of the checkpoint inhibitors. And so I think that's really what we do when we do science. We never really understand. It's very hard to predict fully what the implications are going to be. Just knowing stuff, just knowing about the structure of adhesion, of coherent-based junctions or their sensitivities to tension and structure, provides a little entryway into maybe knowing something else and maybe knowing something else. That's my answer. I'm sorry. Yes?
Thank you. I want to reply on the question, the former question.
Yeah.
In cancer, it is not a myosin who will be the root factor.
Yeah.
We have a very, now with the CAR T cells,
Yeah.
we know that we have to block certain things. And one of them is even myelin, because we get from this kind of cell, mature cells, senescent cells,
Yeah.
we go to zombie cells.
Ah.
If we
Yeah.
go to the telomeres and we stop there, we're going to have a big problem with this kind of acids.
Yeah.
And hyaluronic acid, I block a lot of acids in the membranes of the cell. And now my question to you.
Yeah.
Do you see myosin as an enhancer for cell communication? In a way that when goes it over to the calcium diaphragms and the cross-talking cells?
Yeah. So I think my problem with myosin, even though I'm going to be I'm the myosin biased person, myosin does everything, but that's probably the problem. It does too much. And I can't believe that, so if you look at the immediate consequences of myosin activation, it's a restructuring of all the junctions, adhesive junctions, and we know that many signaling molecules are there and are being restructured as well. All of if I were to use antibodies to the Notch receptors or other basic signaling pathways, they cannot avoid being affected by myosin and so it's I said that was why the gist of my answer effectively is that I think the only value in the results that I've told you is that they point to some interesting aspects of structure and function and cellular transitions that might provide openings for a more sophisticated analysis. And I think we have to deal with signaling and have to deal with we have to deal with intermediate states and zombie cells, if you will. Right, yeah.
Hello, I would like to ask a question. So, what is first your theory about change in the localization or distribution of the myosins in the cell when it shifts from the apical to basal, I don't remember, and whether you were considering that change in the cell shape leads to change of intracellular distribution of the protein producing machinery,
Yeah.
and whether this can have a consequence of how efficiently and rapidly the extracellular matrix and fibrillary proteins can be, you know, changed within the
Yes.
cell.
Okay,
Thank you.
so I think I'm not fully confident in what I'm going to tell you now, but our sense, and I may have misspoke when I said that myosin, you see changes in the distribution of myosin, and its accumulation basally and its accumulation apically. What I don't think really happens is that myosin that was in the basal part of the cell gets redistributed to the apical part. I think there are large cytoplasmic pools, and we're essentially observing local activations. And so it's not so much a change in distribution strategies on this short time scale. I think that in the longer time scales, things like that can clearly matter. But there's also some genetic experiments, and we don't understand mechanistically or genetically what's driving these two things, but we do know that we can block the Rho pathway, this activation of apical myosin, and the basal, so that you don't accumulate it apically, but the basal loss still occurs. So the two phenomena can be uncoupled genetically. And so we don't think that, we think in a way it's even useful that they are independent for controlling the morphology. But again, that's a speculation done on not really complete experiments that are not really fully understood.
Okay, yeah. I just have maybe a brief question. Like you were having the slide where you were pulling with the magnetic field, and you were talking that gradually the tension was lowering as you were pulling, so it was not a perfect spring.
Yeah.
And I was wondering, like, were you looking at the different times and forces applied? Because you said like there is no actin broken or...
Yeah, it's...
Because I was just thinking, if the spring cannot be just the elasticity or like elasticity of the cell membrane that you are seeing.
Right. So it's dependent on actin, the behavior. We don't know where the spring is. We don't, and there's an even deeper danger in the logic in that intuitively, with my limited memory of physics of springs, we think that you stretch a spring and you store potential energy and the spring comes back unless you disrupt the molecular. And so it's a simple process. What we see in flies is a phenomena that behaves like a spring in that it remembers a resting length and drives back to that resting length. I haven't really shown you that you could, I haven't really shown you that this is due to a spring. What I've shown you is that it's due to a phenomena that is like a spring and it has those properties, and then you go back. Now, very possibly, the experiments that you see, we were more clever in our use of distortion and our use of the magnets. We could begin to ask, how much like a spring is this really? Or is it that cells remember resting lengths not because they're like springs, physical springs, but there's another memory device for resting lengths, and you achieve that length, and it looks like it's a spring. You always go back, you stretch it, and then you always go back, but cells are burning up energy to go back rather than using stored potential energy. I don't know if that was a clear answer, but those are many of the things we don't know. Thank you.
Thank you. Okay. There are no more questions. Actually, I would like to wrap up the session because I just realized a horrible thing I did at the beginning. because I stopped, when I was talking about Eric's career, I stopped in the Basel years. I forgot to tell you that after that, he went to Zurich for the postdoc, then he got a first independent position at EMBL in Heidelberg, and then he moved to Princeton, where he is ever since.
Yeah.
And I think he's like one of the few Nobel Prize winners I introduced without ever mentioning he got a Nobel Prize.
Oh, that's okay. I didn't always have a Nobel Prize, and it was, you know, it would have been, you know, it's great to be a scientist. You have to approach the science. The reward is being a scientist. The reward can't be getting a Nobel Prize. I think that overall it's a good idea to recognize scientists and recognize great experiments. And I think it's good for the public. The public takes that seriously. I think it's something that you know, and it's not just because I benefited from it, but I think that it is a good idea. But for any one of us individually as scientists, we better well have our life organized in a way where the reward is someplace else. Because the reward really should be that you do science.
Thank you, I feel much better.